This is a FarmDoc daily webinar. I'm University of Illinois Extension's Todd Gleason. Today we'll take up crop sciences, in particular weed sciences, and talk about water hemp and the fight against herbicide-resistant water hemp. We're joined today by our presenters, including Aaron Hager and Travis Wilkie. You may always find our programs online easily enough at the FarmDoc Daily website, farmdoc.illinois.edu after the fact. If you'd like to look at this or other webinars, they're all under the webinar section. Good morning to you, Aaron and Travis. Thank you for being with us. Aaron Hager, I think you're going to kick us off for the day.
I will kick us off, and then toward the end, we're going to turn it over to Travis Wilkie, who will share a little bit about the research project that he is about to start for his master's degree here at the University of Illinois in the Department of Crop Sciences. But I was thinking back earlier this morning when I first put together this particular slide and this question that we call the water hemp conundrum. I think I actually put this together and started using it about 15 years ago. And for probably 12 of those 15 years, I really just asked the first question, and that is, how do you manage a population that there may not be any viable post-emergent herbicide options for its control? But unfortunately, as we've seen water hemp continue to evolve and raise new challenges associated with resistance, we had to add the second part of that question, and that is, and reduced residual control from many soil-applied herbicides. And that, of course, is going to be one of the more significant challenges of the type of resistance that we will talk about here this morning. Now, we always come back to this slide also because it's very, very important when we talk about water hemp, because we really have to understand the biology. This is really the key. It's the key to understanding why this continues to happen with these species, but it's also the key to how we ultimately can be successful in our fight against herbicide-resistant water hemp. And of course, the first thing, and one thing that's very important with respect to what we'll talk about toward the end, and that is collecting seed from water hemp, is the fact that we're dealing with what we refer to as a dioecious species. And all that means is that any water hemp field or any water hemp plant growing in a field is either a male or a female. All the male plants do is produce pollen. All the female plants do is produce the seed. But in order for that seed to be produced, what has to happen is that those female flowers have to be pollinated. So it's an obligate outcrossing species. But the catch, so to speak, is that there's no biological law that says all of the flowers of the female have to be pollinated by the same male. It doesn't have to work that way. So in other words, if she produces, let's say, 500,000 seeds, that means she has 500,000 flowers. At least theoretically, she may be pollinated by as many as 500,000 different males. So that leads, of course, to a lot of diversity that we see in water hemp. And the other very important characteristic is that the female plants can produce a tremendous amount of seed, a large amount of seed, several 100,000 seeds per female growing in competition with corn or soybean. That's not uncommon. It's actually been documented in the literature that some female plants growing in isolation can produce in excess of 1 million seeds per female. And unfortunately, one of the ways that we see this tremendous amount of genetic diversity being exhibited in these populations is in the continued evolution of resistance to various herbicides. Here in Illinois, we actually now have confirmed resistance in Illinois water hemp populations to herbicides from 7 different site of action groups. Prior to about 2012 was the first time that we actually identified and made confirmation of our first population that exhibited resistance to the group 4 herbicides, things like the 2,4-D, and most recently we have confirmed dicamba resistance as well. But the focus of what we're here to talk about today is actually the last group or the most recent group that we've identified, and that's what we call the group 15 inhibitors. So it's been now about 4 to 5 years when we first made the confirmation about resistance to group 15 herbicides. And it's been very interesting because this is a very old class of chemistry. It's actually been first discovered clear back in the 1950s. These products are somewhat unique in that they have really only pre-emergence activity. They really don't have any foliar activity, so this is why, of course, most of these are going to be sprayed and come in contact with the soil. Now, when you see the group 15 or the VLCFA, that stands for very long chain fatty acids. And so these herbicides actually target these VLCFA elongases. And subsequently, what happens is essentially the plants starve for these long chain fatty acids. And these are very important for various plant components, for example, for cell membranes, as well as waxes on the leaf surface. And generally, a lot of times when we use these products, a sensitive plant will either fail to emerge from the soil, or if it does emerge, it will remain in a very, very small state, arrested growth state, and probably not even visible unless you get down and look very, very closely. So these actually are the Group 15 products that we're talking about. These are the ones that we commonly use here in Illinois and certainly across many areas of the Midwest. Product names like Harness and Warrant, which contains the active ingredient of acetochlor. We have Outlook, that's marketed primarily by BASF, Dual 2 Magnum. And actually the newest Group 15 herbicide on the list is Zidua. So we said these are old chemistries, and they certainly are, been around again since their first discovery back in the 1950s. But their importance in weed management remains very, very high. For many early years, when using Group 15s, these were primarily the soil-applied herbicides that we used for grass control in corn. But over time, and certainly as we've seen the evolution and the expansion of water hemp across the state, these Group 15 products have also become now very important for control of these small-seeded broadleaf species. If you would, pull up a dozen different herbicide labels and that contain Group 15 products, a lot of these products you could either apply by themselves, or there's a tremendous number of premixes that have one of these Group 15 products in here. And in the last probably 5 to 7 years, we've seen a lot of momentum being gained around this idea of layering residuals, especially in soybean. So we would make an application of a residual product close to soybean planting time. And then when we would come back for that first post-application to control any emerged weeds, common practice now is to add one of these Group 15 products in there with the idea of trying to extend this residual control for several additional weeks into the season. And if there's good news around this, of course, it's that really, relatively speaking, resistance is rare to the Group 15s. There's approximately somewhere around 13 species worldwide that have evolved resistance to Group 15 products, and only 3 dicot species. Now, the bad news is 2 of those 3 dicot species are amaranthus. One, of course, is waterhemp, and the other is Palmer amaranth. And so how did we find this? How did we first stumble into this idea of Group 15 resistance? And to be completely honest with you, we actually got our first inclination or first feeling about this, not by a failure of a Group 15 product, but actually the failure of a Group 27. And what this slide represents, this is actually a photograph that was taken clear back in 2010. This is actually the first water hemp, or actually the first weed population anywhere in the world that had evolved resistance to the Group 27 herbicides. In particular, this was mesotrione that we're showing here. But we had several experiments on this location, and we noticed when we looked at some of these soil-applied treatments that we had, of course, across the bottom of this table, you see the non-treated. That obviously did not have any herbicide applied to it. We're looking at the columns here. We'll look at the columns of control at either 30 days or 60 days in 2010 or 2011. And right above the non-treated in the table is S-metolachlor. And that's a very common field use rate. But what we noticed is that the level of control of this particular population was much lower than we normally associate it with. Now, many weed scientists would probably agree that acetochlor tends to be a little bit stronger on some of the smaller seeded broadleaf species, but certainly not to the degree that we were seeing in this particular population. But that was really not the focus of that work. We were focusing more on identifying and understanding the Group 27 resistance. So we sort of just set these observations to the side. We knew they were there, but that really wasn't the focus of what we were following up at that time. Well, sure enough, a few years later, we had a second population here in Champaign County where the farmer had used some of the Group 27 products and had really not had very good efficacy against that population. And so fortunately, we were able to gain access to that field. And lo and behold, my student at the time, I asked him to put on about 3 rates of 2 or 3 different group 15 herbicides and tell me what did you see about this? Well, about a month and a half later, he came into my office, and what he described was almost identical to what we had seen from the group 15 performance at that McLean County population. And so we were on to something here, at least we thought we were on to something, and we wanted to investigate this a little bit closer to see do we actually have a resistance now that's evolving to these group 15 products? And I won't bore you with a lot of data about this, just a photograph here I think would probably be sufficient. This is actually a field photograph that we took at 28 days after these group 15 products had been applied, and the year was 2020. Now, across the bottom panels, that is a sensitive population from Urbana. Over here on the far left, you'll see the non-treated. And then in each of these individual panels, you'll see what the group 15 product was and the application rate. And if we look across here, really, we don't really see anything that surprises us here on this sensitive population from Urbana. But if you look now at the panels across the top of the slide, this is a separate population. This is the suspect or now confirmed group 15 resistant population. And you can see that the level of control of this population, regardless of which group 15 herbicide we're looking at, is much less. Now, one thing that we've learned, of course, over time is that we really can't make a pronouncement or confirmation of resistance based only on field work. We have to go into the greenhouse and do a little bit more detailed work. In particular, we do what we call dose response experiments. And the advantage of doing this in the greenhouse, of course, is we can control and or eliminate a lot of the environmental variability, a lot of the soil variability, and we can compare the responses of our suspect population to the responses of a known sensitive population. And so this is just basically the basic materials and methods that we use in here. That's really not important. But what we typically do in dose response experiments is that we will use treatments that range from an extremely low rate to an extremely high rate. And just to give you an example, if you look here on this particular slide, the rate structure that we use for dual tube magnum, the low rate was about 1/8 of a fluid ounce. The high rate was a gallon. So obviously we were at a low rate that was so ridiculously low, we didn't really expect it to do anything, clear up to a rate well in excess what's labeled for use in the field. And this is what we see. And I'll set this slide up a little bit because the next couple of them are going to be somewhat identical to this. These are the results that we see at 21 days after treatment, after these individual soil containers had been treated with dual 2 Magnum. Now, there's 4 populations represented in this slide. If you look toward the top, the very row across the top is labeled CHR-M6. That was one of our suspect resistant populations. The row of plants immediately underneath that, labeled MCR-NH40, that was the field population from McLean County, where we had first seen that low level of control with some of the group 15 herbicides. The lower 2 rows are populations labeled WUS and ACR. Those are 2 of our sensitive populations. And so now if you look vertically at the column, so the 4 pots from the bottom to the top of the slide, you can see what the rates are. So clear over on the left, all those 4 pots that are labeled 0 were not treated. And then you can look at the individual rates for each of those columns of pots. The one labeled at 2,700 grams per hectare, that is roughly about a 1x rate, or what we would consider a 1x use rate of dual 2 Magnum in the field. So what do we see here? Well, if you look again toward the resistant populations, the CHR and the MCR, at that field use rate, we still have plants that have emerged through that treatment. Matter of fact, if you look very closely, even at 8,400 grams, a 3x rate, you can see that some water hemp actually had in the MCR population emerged. But now look at the 2 sensitive populations. Really, we see good control of these 2 sensitive populations at only 270 grams. So really at a 1/10 of a field use rate, we're still getting good control of the sensitive populations. But even at a field use rate or 3 times the field use rate, we're seeing emergence of the resistant populations. Now we do these various rates so we can generate the curves that you see over here on the left hand of this slide. Okay, so across the bottom of each of these slides on the x-axis, that is the rate of the S-metolachlor. The bottom graph labeled B, that is calculating survival of these populations. And the upper panel labeled A, that is the dry biomass of these populations. Now, from these curves, what we're trying to do is determine what rate of S-metolachlor reduces one of these growth parameters, either survival or dry biomass, by 50%. And so now let's look over at the actual table. You'll see in the first column the 4 populations that are listed there, the CHR, the MCR, the resistant populations. The 2 sensitive ones are labeled WS and ACR at the bottom of the table. The important column I'll draw your attention to is the one labeled the LD50. That is the dose or the rate of herbicide required to reduce the emergence or survival of these populations by 50%. We get that from looking over here at these curves. We go up to about 50% of the control, draw a vertical line over to our populations, and then drop straight down to get what those rates are. So let's walk through this. For example, the ACR, one of our sensitive populations, it took only 53 grams to reduce that population by 50%. The other sensitive, WUS, it took 101 grams. Now, if we go to the top of that column, the CHR population, it took 1,800 grams. And so what do we do with these numbers? Well, if we take 1,800, for example, which is the rate for the resistant population, if we divide that by 100, which is the rate for the WUS, you get a value of 18. That you'll see in the third column as the R to S ratio. Those ratios essentially give us an idea of how resistant these populations are. And we have seen these ratios range anywhere from single digits all the way up to several thousand-fold resistant. So how would we use this information? Well, for dual tube magnum, based on this work in the greenhouse, we can say that the resistance ratios of these populations range from 18 to 64. And that all depends upon which one of the 2 sensitive populations to which we're drawing the comparison. So what about the other group 15 products? We see the exact same trend. So now we're looking at the results 21 days after acetochlor or harness had been applied to these. Here, the 1X field use rate is clear over to the right at 2,500 grams. But look at the pattern of the emergence of the various populations. So we can still see relatively good control at only 10% of that rate on the sensitive populations, but the resistant populations have still emerged. And so we are able, of course, to generate curves, and from these curves we can determine these LD50 values and these resistance ratios, these R to S ratios. So you've seen what we have done now for S-metolachlor, what we've done for acetochlor. We see the exact same pattern for dimethenamide or Outlook, and we see the exact same pattern or this rate shift, so to speak, with Zidua. And so once all this work was done and compiled, here's what we found out based on the ratios in the greenhouse. The reason that we think this is very important to emphasize this, if you remember back to the slide that we looked at the field results and the 4 panels of the sensitive and resistant populations, it looked like, and I believe rightly so, that there were fewer water hemp in the acetochlor treated plots relative to the other 3 group 15 products there. But to say that the simple solution to group 15 resistance is to just rotate, say, from a metolachlor product to an acetochlor product, that's missing the boat. And it's missing the boat because it's resistant to not only S-metolachlor, these populations are resistant to every one of the group 15 products that we've looked at. What's different among the group 15s? It's the ratios. So we tend to see the highest resistance ratios to the S-metolachlor products, the lower ratios to either peroxysulfone or acetochlor. But the key message, folks, is that the resistance is there to all of them. So the first ones that are going to emerge through an S-metolachlor treated area are going to be the resistant ones. The first ones that are going to emerge through an acetochlor treated area are also going to be the resistant ones. So just to wrap up a little summary here on this part of the presentation, what we haven't shared with you is the additional work that we've done with these populations, and essentially what we know now is that the mechanism of resistance, or the way that these plants or these populations are using to survive these group 15 herbicides, is what's called enhanced metabolism. So in other words, the plants, the seedlings are actually taking up the herbicide, it's getting into the plant, but they've evolved mechanisms now that allow them to rapidly break down that herbicide before it can actually cause a lethal effect to the plant. The scary part, or perhaps the unnerving part, is that these water hemp populations now are using the same technique that many crop plants use in order to survive being treated with a herbicide. The reason that we don't control corn with atrazine is simply because the corn can rapidly metabolize the atrazine. Now we're seeing more and more of these populations that are able to break down some of these group 15 herbicides literally as rapidly as corn can. And rapid metabolism is the mechanism by which corn is resistant or tolerant to these group 15 herbicides. Now, we won't go into all the details about this, but what we really want to emphasize now is that this is continuing to happen. And any water hemp plants that have survived the 2023 growing season, and any females that now have seed produced on those females, have gone through one additional season of selection. We've treated them for whatever reason they have survived. So maybe those 5 or 10 individual female water hemp plants that you've got across that 30-acre field, they're not going to slow up the harvest at all. They're not going to plug up the combine. But what happens if some of those seeds actually now have a new resistance mechanism with them? And now all of a sudden, we're going to allow that seed to now enter into the soil seed bank and be part of the problem for future years. So some of the implications of this, we have confirmed this now in 2 populations, but we really have no evidence to assume that it's in only 2 populations. We really don't know how common this is. That's what we're here to try to find out. How actual common is this? One of the most significant implications of this work is that this resistance now, the way that this resistance is manifest in the field, not necessarily the greenhouse or laboratory, but in the field, in farmers' field where these products are used, is that the length of residual control now gets less and less. So say hypothetically, we would use a soil residual herbicide on sensitive population that gave us, let's just say 4 weeks, perhaps 5 good weeks of residual control. If we would use that same active ingredient on a confirmed resistant population, maybe that length of residual control is now only 3 weeks. Maybe it'll be 2 weeks. Maybe those survivors in the field right now have a different mechanism. Maybe now some of these will only be a week. So that's really the concern about this. If our residual control is less and less, that means that we may have to think about moving up the timing, literally on the calendar, of our post applications, because our length of residual control is simply not going to be as it once was on sensitive population. And remember, we talked about this idea of this overlapping residuals at the beginning of the presentation. The overlapping residuals that we use in soybean are all group 15 products. So that really is something that we need to be aware of, that we may not be able to extend that length of residual control following that in-season application of our group 15s. We still think, and we still encourage people to use multiple effective herbicides. In all honesty, we think that's the best recommendation, but I have to admit that we really don't have a lot of data to support that anymore. We really need to think about the idea that we're going to continue to treat virtually every acre of corn and soybean, not only in Illinois, but perhaps many areas of the Midwest, with herbicides. But because of this continued evolution of resistance, we really have to have a serious conversation about what else can we do to try to ensure that by the end of the growing season that these female plants have no seed. Because amidst all this uncertainty that we have around, well, which herbicides may work yet, which ones may not work yet, or do these females in the field right now contain the next generation of resistance mechanism? The only thing that we really know is that if there is no seed, there's no change in the frequency of any of these resistance mechanisms. That's really all that we know right now. So again, we talked about the biology of water hemp at the beginning of the presentation. We said that's really the key. And the key really is that we're not necessarily going to wait and open up a new jug of herbicide that the industry brings us at some point, because that's probably not going to be very sustainable. But what we do know is that the weakness of water hemp, the weakness of Palmer amaranth for that matter, is that the seeds do not remain viable in the seed bank indefinitely. And so these other practices, whatever they are, maybe it is picking up a hoe or a weed hook and walking a bean field. Maybe it is the idea of harvest weed seed control in the back end of a combine. Maybe in some cases it's the electrocution of the weeds after they get a height differential over the soybean crop. Any of these things that can reduce that amount of seed, that's the win that we need to look for. Okay. And of course, we want to just caution folks that not every time that we see a failure of a field treated with a herbicide is not always caused by resistance. So I guess we go back to one of the initial questions that we had. How widespread is herbicide resistance, in particular, resistance to our Group 15 products? We know of two populations, one from McLean County, one from Champaign County. But as we mentioned earlier, we really have no evidence that those are the only two populations. And so we'd like to thank the Illinois Soybean Association for providing some funding that hopefully is going to allow us to have a better estimate on how common this is. And with that, I'm going to turn it over to Travis Wilkie, and he's going to share with you a little bit about what his project is going to be here, working on his master's at the University of Illinois. So Travis.
Thank you, Dr. Hager. Yeah, so here in the next couple of weeks, I'm really excited to kind of start going around the state and making some of those random collections for water hemp to be used later in those screenings and potentially in that dose response survey. But with also in that project, we're very excited to be able to test the public submissions that are brought in. So the video that I'm about to show that's on the farmdoc YouTube page gives some really good guidance in how to make those collections and send them in. So I'm going to pull that up really quick. Hello, my name is Travis Wilkie, and I'm a master's student under Dr. Aaron Hager in the Weed Science program at the University of Illinois. I want to thank you for your interest in submitting samples of water hemp for this Group 15 resistant survey. And I also want to give a big thank you to the Illinois Soybean Association for providing funding. The purpose of this video is to help us differentiate the water hemp from similar species and provide instructions for submission. So the first thing we want to talk about is differentiating water hemp from similar species such as smooth pigweed or red root pigweed. The best way to do this in the field is to look for hairs on the stem and leaves of the plant. If those tiny hairs are present, you're probably looking at smooth pigweed or red root pigweed, and we do not want these weeds for our samples. Water hemp, on the other hand, has an absence of hairs on the stem and leaves. With that being said, we also have to differentiate within the water hemp species itself. Water hemp is a dioecious species, meaning that we have male and female plants. We need to collect the female plants to make sure that we actually get seeds and not a wasted submission. The best way to do this in the field is grab some of the inflorescence of the plant, simply just the flowering parts, and crush them in your hand a little bit. If you begin to see small black to dark brown seeds, then you have a female plant, whereas if you're only getting flower parts, you have a male plant, and we do not want that submission. Whenever we collect these, we want to avoid the edges of the field as much as possible and make sure that we're only using one female per bag and that those two females are 30 feet apart. So whenever we go to making these submissions and putting the plant in the bag, we need to keep in mind that we need as much seed as possible. So fit as much of the plant into that paper bag as possible and use only paper, as plastic will preserve the moisture and jeopardize the chances of getting viable seed from that plant. If you need to cut the plant up in any way, please do so by maintaining the upper half to third where most of the flowers are at on the plant to make sure that we get as much seed as possible. Once you have selected your female water hemp and placed it in the bag, staple it shut with the submission form that is provided, and repeat this with another female specimen at least 30 feet away, and attaching the second submission form to that bag. I'd like to thank you all for your interest in this project, and another thank you to the Illinois Soybean Association for providing all of the funding. If you have any questions, feel free to reach out to myself with the information provided, and I wish you all the best in collecting your water hemp samples. So like I said earlier, that video is still on the farmdoc website if you need to refer back to it. But I think that's where a lot of the excitement for myself comes from this project, and comparing the resistance that we find from, like I said, randomly collected samples versus those that are sent in by people around the state, potentially already having that idea that they're looking at a resistant population. We've talked a lot about how these submissions can be affected by the viewpoints that we have earlier in the season, such as a failed application, whether it be from not having enough rainfall on those group 15 herbicides, or if we just have other soil factors that are coming into play. So this goal of that public submission side of the project is very exciting in the fact that we can narrow the problems that we would expect and kind of cancel out all of those problems that we might have in the field, and be able to tell for sure if we're looking at true resistance or not. So with that, there's also a sample submission form that needs to be sent in that can be found at the QR code on the screen or that URL. Please follow all of the directions that are on that form when stapling it to your bag of water hemp seed, and we'll be able to either accept them in person, or those samples can also be mailed in. But I think that kind of covers the public submission side of that. So along with the public submissions, we'll be looking at least 100 different randomly collected samples from within our department, as we'll be around the state collecting those and making sure that we stay ahead of the combines this fall to get those samples in as well.
If you have not gotten that QR code yet, don't worry about it. It'll come back up in just a second. If you've got questions for Aaron and or Travis, do write them into the question box that's over in the GoToWebinar control panel. Those are the gray boxes that are either at the left-hand side of your page or down at the bottom of the page. We would like to thank our sponsors for the FarmDoc webinar series. They include the TIAA Center for Farmland Research, Compere Financial, Corteva, Illinois Corn Growers Association, Growmark, Farm Credit Illinois, and of course the Illinois Soybean Association, along with our educational partners, FBFM, that's the Farm Business Farm Management folks, Illinois Extension, and of course the College of Agricultural, Consumer, and Environmental Sciences, Department of ACE, that stands for Agricultural and Consumer Economics. You can always find this webinar and many others online at farmdoc.illinois.edu in the webinars section under archive, or just go straight to youtube.com/@farmdoc. And of course, we'll bring up that last slide too, which hits you back to that QR code if you're interested in wanting to make sure you have access to the submission forms. go.illinois.edu/FD-waterhemp. A couple of quick questions. Aaron Hager, for those producers and others that know they probably have resistant water hemp or think they do, along with making the submission, what are the things that they should do at the end of the season or as they run the combine through the soybean fields to make sure they're not spreading that water hemp from one place to another?
I think one very important thing, Todd, is to, try to avoid running the combine through these very heavy, thick patches. Say if we had a drowned out space, which this year, unfortunately, we don't probably have as many drowned out areas in fields as what we can in a normal year. But the other thing to think about is, can you prioritize the sequence of fields where you do harvest, and try to save the fields that have the heaviest populations of weeds, whether it be water hemp or any other weed for that matter. Try to maybe wait and harvest those last, because as soon as we run these, weed seeds through the combine, combines are one of the most efficient weed seed spreading operations or machines that we have. And we will move these seeds around from that point on to any other field where that combine is going to be used this fall.
And Travis, as you were explaining in the video how to recognize the proper water hemp species, I know you've talked about the hairs on the plants for the red root and others, and not on the water hemp. What are other common mistakes that people make when they're trying to send in these particular samples?
I believe probably one thing that we definitely want to recognize is that we're going to need as much seed as possible. So, along with making sure that you recognize the correct species, we can't stress enough how important it is to get a large amount of seed in that bag. Um, so, while we will be stratifying and going through a lot of processes to make sure that we get as great as germination rates as we can in the greenhouse, we still want to make sure that we're having as much seed in that sample to test as possible. So, um, once you start with just recognizing that it is truly water hemp and not another similar species, and making sure that it is also a female plant, um, we really need to make sure that we're getting a large amount of seed, and that's definitely something I would stress to anyone who's sending in submissions.
Do you have guidelines for what that means? And typically, a single water hemp plant will have many seeds? When large amount may be a variable here that is really different than folks might expect. And are we talking you want to collect 3, 4, 10 plants, one plant, and again, how many seeds per plant might there be?
Actually, Todd, what we're looking for and what we've had in previous years is that people will clip one little inflorescent stalk off a female and send it to us. That's really not enough. Unless you really have a deep desire to keep these females in your field or in a field, send as much of that plant material to us as you can stuff into that bag. That will help ensure that we have enough seed that we can get to germinate and emerge in the greenhouse and be able to do these screens. The reason we ask only one female per bag is because it goes again back to what we talked about, the biology. It's a dioecious species. So all the seed on any one female, all we know for sure is that they all came from the same female. We have no idea if they all came from pollen from the same male or from hundreds of different males. So by trying to reduce some of that variability by only putting one female in a bag, it helps us to really reduce some of the initial variability that we see in these early greenhouse screens.
We, of course, have been talking with Aaron Hager, who you just heard. He's a weed scientist with University of Illinois Extension, located here on the Urbana-Champaign campus of the U of I, along with a master student in weed science, Travis Wilkie. Our program was produced with technical assistance today by Jim Bolts. I'm University of Illinois Extension's Todd Gleason. If you'd like to see it again, you can always do that on our website, farmdoc.illinois.edu, and then look for webinars and archive, or go to youtube.com/@farmdocvideo. You have a good day. I'm Illinois Extension's Todd Gleason.